So for the next discussion is we will going to talk about the protein as an enzyme. So before we get into it, let's define first what is a catalyst. Talking about a catalyst is a substance that speeds up the reaction. So it can either be inorganic or biological, or biological catalyst is also known as enzymes. So catalysts basically they speeds up the reaction, but in the end product they are not consumed during the reaction or speeding up the reaction of the substance or forming the product. They are not consumed, so they can be recycled. So they are very important in the biological system, since sometimes reactions could take millions of years to produce a product. So with the help of catalyst, they lower that time rate. So later on we will discuss on how scientifically it happens. So let us take note that not all catalysts are, or not all enzymes or biological enzymes are proteins. There are some exceptions like ribozymes. Later on you will know it in the central dogma of biology. So now that we know or defined what is an enzyme and catalyst, let us talk about thermodynamics versus kinetics. These two concepts are different, since they can either be correlated or not. So, but I'm sure that these two, please do not be confused on these two concepts right here. When we talk about thermodynamics is talks about whether or not the reaction is spontaneous or not. Or will it occur or not. So, ganun lang yung thermodynamics. An example of thermodynamic reaction or thermodynamically favored reaction is combustion of diamond to form carbon dioxide. And for kinetics, this tells us how fast the reaction is. Example is decomposition of hydrogen peroxide. Now, why did I say that we should not be confused on these two terms? Sometimes there are reactions, especially in chemistry, that is thermodynamically favored but kinetically not favored, and kinetically favored but not thermodynamically favored. So in the example shown here, the combustion of diamond to form carbon dioxide is thermodynamically favored but kinetically not favored kasi matagal siyang parang gawin, or matagal siyang parang in the process, matagal siya. But in below naman, makikita natin kinetics, or the example of kinetics here is the decomposition of hydrogen peroxide. Kinetically, this is favored kasi when you put hydrogen peroxide, it will easily evaporate. But kinetically, in terms of energy, no, it is not really kinetically favored. So I hope that's clear. So now that we know the concepts of thermodynamics and kinetics, now let's have this graph. So, this is the graph that shows how an enzyme, or the difference between a reaction with catalyzed reaction and a non-catalyzed reaction. So here, when this flow, parang itong curve na ito, this talks about the uncatalyzed reaction. So, as you can see, mataas siya. And since mataas yung curve niya, matagal yung reaction rate niya. While here, yung parang konting curve is the catalyzed reaction. As you can see it, maliit lang siya. So, take note that despite catalyzing or uncatalyzed or catalyzed reactions occur thermodynamically, or when we talk about thermodynamics and kinetics, enzymes, despite being speeding up the reaction, it will never change the delta G. What is the delta G? Delta G talks about the Gibbs free energy change. So, this talks about the change in energy. Take note na hindi catalyzed man or uncatalyzed, it will never change the delta G. So, how does it happen? So, taking into account the concepts that enzyme do not change the standard free energy change, or the delta G, or the free Gibson energy change, what does it mean? No? Kasi, di ba, speed up ang reaction pero hindi niya change ang delta G. How does it occur? Or how does, paano siya? Paano siya possible? So, ang analogy is that, it is like you are going, let's say, you're going to Davao, no? There are a lot of routes or ways that you can take. You can take a longer route or shortcuts. But in the end of the day, you will still arrive at the final point. So, catalysts, like the analogy given, no, they find a alternative route, but ultimately, the initial and final point of the trip is the same. So, take note. Tingnan natin dito. Ito ang initial point natin, and this is the final point. Look at the graph below, above, no? Catalyzed man or uncatalyzed, they still end up at the same point. Kung i-measure natin ito, yung distance dito until dito, it is still the same. They both arrive at the final point of the destination. Kaya, sinabi sa, if you have the Campbell biochemistry book, it says there that catalyzed man or uncatalyzed, it never changed the delta G. Kasi they will still arrive, they came from the initial point and will still arrive at the final or the same final point. Kaya, enzymes do not change the delta G. So, now that we have established the difference between kinetics and thermodynamics, we also now determine what is a catalyst, no? Let's have first a short review lang before tayo mag-proceed sa enzyme kinetic equation. So, as we all know kanina lang na catalysts are any substance that speeds up the reaction. It can be biological or inorganic. Biological catalysts are also known as enzymes. They speed up the reaction by lowering down the activation rate, activation energy. In return, despite being speeding up the reaction, they are not consumed in the process. Kaya recyclable ang mga catalysts or mga enzymes natin. Again, we discussed the term what is thermodynamics. They talk about how spontaneous the reaction is, and the kinetics or tells you how fast the reaction will happen or how fast the reaction. So, let's talk here about the enzyme kinetic equation. So, we can express the enzyme kinetics into an equation. Above there, as you can see ito, sa taas, no, it is the simpler version of the kinetic equation or the enzyme kinetic equation, wherein A plus B will form P. So, as my former professor said, na the simpler, the wronger. So, masyadong simple ito and not so detailed. So, let's complicate things a little bit more. So, let's elaborate further this equation into this. So, we can say that the rate is just equals to the negative delta concentration A. Take note, bakit naka-bracket si A, B, and P? Because it talks about the concentration or the molarity. Concentration yan siya. So, pagkabasa nito, negative delta concentration A all over delta T is equals to negative delta B, concentration B, all over negative delta T is equals to delta concentration P all over delta T. Wherein delta T talks about the time. So, bakit ba negative si concentration A and concentration B while concentration P is positive? The reason is that negative delta A and negative ito silang dalawa kasi it talks about the disappearance, and positive si P kasi it talks about the appearance of P. So, how does can you, if you were asked to me na can you elaborate, no? So, since ito, given na ito na equation, we can say that the concentration A and B disappears automatically, P will appear. Or in other words, the rate of reaction at a given time is proportional to the product concentration of the reactant raised to the appropriate power. So, ganun yun siya. So, with that being said, this equation can actually be written as also as the rate infinity concentration A raised to the power of F, concentration B raised to the power of G. Kasi sabi dito that raised to the appropriate power. So, where F and G are the exponent, or technically speaking, F and G can be determined experimentally. So, we can say also that this equation, rate infinity, concentration A raised to the power of F, concentration B raised to the power of G, we can say, or we can rewrite this as the rate is equals to K, concentration A raised to the power of F, concentration B raised to the power of G. So, wherein K is the constant or the coefficient of our equation. So, ganun yun siya. So, I hope that is very clear. So, lastly, let's talk, before we end this video, let's talk about the temperature and pH. So, how does temperature and pH level affects the enzyme's effectiveness? So, ang answer lang nito guys is simple. Let's talk about first with temperature. Temperature, higher temperature, faster performance. So, kasi bakit faster performance? As we raised up the temperature in our system, there is a lot of energy available for the enzyme to use. Kaya faster ang reaction rate niya. So, but warning, too much will denature the enzyme. Bakit? Let us remember again that enzymes, or like proteins, yung mga enzymes natin are just made up of molecules. They obey the simple, yung laws of interaction, yung London dispersion, stuff like that. They just obey those rules. And as you probably learned in the previous chapter, temperature actually denatures the enzymes kasi, or the proteins, kasi heat will disrupts the bond in between those interaction, yung mga London dispersion natin, yung mga IMF natin, intermolecular forces natin. Heat will disrupt the intermolecular forces present in our enzyme. Kaya too much of raising up the temperature will lead to denaturation of enzymes or proteins natin. So, pH, in simple terms, optimal pH, faster performance. For example, if a specific enzyme works best at an acidic environment, if you put it in a basic environment, probably pangit yung performance niya kasi they work best at effective, or they work best in acidic, while as compared to basic. Same goes with some enzymes works best at basic condition. If you put them into acidic condition, hindi sila masyadong effective as an enzyme. So, yun lang yung answer na question na ito. So, how does temperature affect the pH, and how does temperature and pH level affects the enzyme performance? Simple, higher temperature, faster performance, but too much will denature the enzyme, and at optimal pH, the faster the performance. So, I hope you learned it. I hope you learned something today, guys. And by the way, stay tuned on part 2 because we will talk about now the Michaelis-Menten equation. So, thank you for watching.